कृपया अपने मोबाइल साइलेंट मोड में रख लीजिए।
हाय उधर
इस कार्यक्रम का ऑनलाइन प्रसारण भी किया जा रहा है, तो लोग इसका फायदा उठा सकते हैं। आज के इस महत्वपूर्ण कार्यक्रम में आप सभी का हार्दिक हार्दिक स्वागत है। तो आज के कार्यक्रम के प्रमुख अतिथि माननीय डॉक्टर मुकुंद गुर्जर जी यहाँ पर आ चुके हैं। जोरदार तालियों के बीच उनका प्रतीक है। सर, आपके प्रति प्रेम का और स्नेह का और आदर का मैं आपको ससम्मान मंच पर आमंत्रित करना चाहूँगी। साथ ही डॉक्टर सी.वी. रमना जी को भी साथ ही मंच पे आमंत्रित करती हूँ और हमारे निदेशक महोदय कुछ ही मिनटों में यहाँ पर ज्वाइन होंगे। जोरदार तालियों से धन्यवाद। जैसा कि हमारी परंपरा है, हमारी भारतीय परंपरा का निर्वाह करते हुए मैं कार्यक्रम की शुरुआत करना चाहूँगी स्वागत से। मैं माननीय रमना सर से निवेदन करूँगी कि एनसीएल परिवार की ओर से डॉक्टर गुर्जर सर का स्वागत करें पौधा देकर। बहुत बहुत स्वागत और अभिनंदन है सर पूरे एनसीएल परिवार की ओर से। हालांकि आप हमारे लिए प्रमुख अतिथि के पद पर हैं, लेकिन हमारे लिए आप अतिथि नहीं भी हैं। आप एनसीएल परिवार का ही एक हिस्सा है। तो इससे पहले कि हम सर का स्पीच सुने, मैं सर का संक्षिप्त परिचय आपको बताना चाहूँगी। हालांकि मुझे अच्छे से पता है कि परिचय देने की आवश्यकता नहीं है। सर बहुत अच्छे से आप सभी को जानते हैं और आप सभी भी सर को जानते हैं, लेकिन कार्यक्रम की औपचारिकता है, मैं सर का संक्षिप्त परिचय आप लोगों को देना चाहूँगी। आदरणीय डॉक्टर गुर्जर सर ने 2 पीएचडी उपाधि प्राप्त की है नागपुर और लंदन यूनिवर्सिटी से, और 1982 में उन्होंने सीनियर साइंटिस्ट के रूप में एनसीएल ज्वाइन किया और बहुत महत्वपूर्ण रिसर्च वर्क अपने आरंभ किए। उसके पश्चात 1986 में वे आईआईएससी हैदराबाद चले गए और तत्पश्चात 1998 में वे एनसीएल के डिप्टी डायरेक्टर और हेड ऑफ द ऑर्गेनिक केमिस्ट्री के रूप में उन्होंने फिर से एनसीएल को पद प्रतिष्ठा प्रदान की और इस दौरान उन्होंने अनेकों महत्वपूर्ण रिसर्च वर्क किए और 260 से अधिक रिसर्च पेपर्स पब्लिश किए, 183 से भी अधिक पेटेंट फाइल किए और वर्तमान में वे, जैसे कि आप सभी जानते ही हैं, कि अत्यंत प्रतिष्ठित एनक्योर फार्मास्यूटिकल्स कंपनी पुणे के चीफ साइंटिफिक ऑफिसर के पद को सुशोभित कर रहे हैं। साथ ही वे बोर्ड ऑफ डायरेक्टर्स के सदस्य भी हैं। इसके अलावा भी उनकी अनेक उपलब्धियां, जिनका यदि मैं वर्णन करूं तो शायद लंच यही हो जाएगा। तो इन उपलब्धियों के साथ आपका बहुत सम्मान करते हुए, और हमारे लिए बहुत गौरवपूर्ण क्षण है कि आज आप हमारे बीच उपस्थित हैं। हम सभी आपको सुनने के लिए सब बहुत उत्सुक हैं। कृपया आप अपना स्पीच प्रदान करिए और मैं रमना सर से निवेदन करूंगी कि सुविधा के लिए आकर बैठ सकते हैं। धन्यवाद।
Thank you very much for your kind words. मैं हिंदी में बोल नहीं सकता हूँ. I would like to thank the director, for giving me the opportunity to speak to you as a Professor Venkataraman endowment as a lecture. Unfortunately, I couldn't meet Professor Venkataraman, although I was in NCL for some few years. But he has contributed a lot, along with Professor Sashadri and Govindachari, Ashima Chatterjee. These are the people who really created the foundation for research, and that foundation, on that basis, we are now been flourishing in our research activities. So my gratitude to Professor Venkataraman for his contribution to science in the country. The topic is something different, normally I give a lecture on one or two subjects or total synthesis, but this is the time I thought, I should find out from myself what kind of science I had done it for the last 50 years. I started my research in November 1974. This November 2024, I will be completing 50 years of my research. How I got fascinated to science, that's the question. Long time ago, I visited my cousin in Bangalore, and her husband was a professor in IIC Bangalore, and I was in school at the time. So there is nothing much to do during the daytime, so I used to visit his lab and see the research scholars working on experiments, discussing the data. And this really fascinated me, and at that time I thought I'm going to do the same thing. So the question was, later on I passed my examination of secondary school, and at that time now I had a problem, what to do next for my future. Most of my colleagues from the school, they joined the engineering courses, because at that time engineering was the most lucrative course, and you can get a very a suitable job, if you have done the engineering course. There was a pressure from my family to do the same thing, and my brother was doing the mechanical engineering from VRC. The issue was, I should do the engineering or I should do something else. But then I saw the courses and the syllabus of engineering. I realized, they teach a lot of things at once, but nothing goes in depth, There's nothing, no subject is taken very seriously, just superficial. And I didn't like that. So I thought I must explore some other scientific endeavors, and I ended up doing BSc from the Institute of Science in Nagpur, and later on I did my MSc from the University Department of Chemistry. And after my MSc, I wrote to my friend Dr. Furness, who was doing his PhD from King's College London, that is there any opportunity for me to do this PhD under the same guidance. I didn't get any later for a long time. So since I had junior research fellowship, so I started doing my PhD under the supervision of Professor Bhagwan at the Nagpur Institute. Days passed, and after a year or so, I got a letter from Furness that Professor Hoff, who was his supervisor, said that he is getting some funding for his for the PhD program, and my name is being considered for that. So I went to Professor Bhagwan and said that, I got a fellowship, so I want to discontinue my PhD here in Nagpur Institute. So he called me, he said that since you have done your work in MSc and you are continuing the same thing here also for PhD, I just want to know how much work you have done. So I gave a presentation, and he said that this presentation is good enough, you can submit your thesis and go. And that's the way, I completed my PhD there, and I would like to, discuss some work which I had done in Nagpur Institute, which was quite interesting on the reaction mechanism. So let's do the science first now. So the PhD program at Nagpur Institute was related to mechanistic studies. When I was doing MSc, there was a request from the botany department that they wanted to have some allyloxycinamic acids. This is this one. These were not known at the time. So I thought it's a very simple compound. You start with allyloxyacetic ester, react with benzaldehyde in the presence of potassium tertiary peroxide. You can get a ester which you can hydrolyze and do the acid. But in the reaction, we got 50% of the acid and 15% of the ester. So we thought that since this elimination of water takes place in this reaction, the water may react with potassium tertiary butoxide form KOH, and that hydrolyzes the ester to acid. So that was a hypothesis we gave, and it was agreed by all of us. But what happened, we had taken several of these allyloxine esters and several of these aldehydes. In all these cases, we got 50% of the ester and 50% of the acid. This was a little bit surprising for us. So in order to prove that this acid is not coming from the ester, we took this ester and treated with potassium tertiary butoxide and little bit of few drops of water, and then worked the reaction in which almost all the ester was isolated. There is no formation of any acid. So we believe that acid is formed by a different mechanism. So I just want to bring to you what is a Stobie reaction. Sorry. Stobie reaction. Stobie reaction is that, like for example, you take aldehyde, dihydrosuccinate, reacted with potassium tertiary butoxide, you get half ester. In this case, you get oxanion, that is the addition of one of the anion here on the aldehyde, and this forms a gamma lactone, and elimination gives you the half ester. So you get acid as well as ester. So we thought whether the same mechanism probably is happening in our case. When you take the aldehyde and allyloxy ester, it forms a carbanion, and there is a formation of this beta lactone, which eliminates to give you the acid. In addition to that, this gets protonated, and you get a base here, so it eliminates also water, you get ester. So this must be the mechanism by which you get the acid. So the Stobie reaction can take place by gamma lactone as well as by the beta lactone pathway. But in order to put conclusively that this is the pathway, then we have to do something more. So what we did is that we took the radioactive benzaldehyde and treated with the allyloxyacetic acid. So we believe that this not radioactive, this isotopic oxygen, if it undergoes this pathway, then there is an elimination of water in which the the isotopic oxygen leaves, and you should not get any activity in ester. But if it goes via a beta lactone intermediate, then you should get the isotopic oxygen in acid. So that's the way we were able to isolate this acid by mass and other means. We proved that the oxygen O19 is present in acid and not in ester. So this is the mechanism we proved beyond doubt that Stobie reaction can take place by a gamma lactone pathway as well as a beta lactone pathway. So what is the PhD program? So when I visited there, there were a lot of work was going on chlorinated sugars. So you may be knowing that the sucralose, which is 1'4,6' trichlorosucrose, was discovered in the same lab where I was working. And there were a lot of, we were asked to make a lot of chlorinated compounds. Some less number of chlorine will give a little bit of sweetness, but if you put a lot of chlorines, then you get bitterness. So then optimum chlorines at optimum position was very important. And you know that 1'4,6' trichlorogalactosucrose became the sweetener. So I think everybody takes it. It's a very safe compound because when Kate and Light, who discovered it, gave it to Johnson, and Johnson did the work with radio-labeled chlorine, and they found out that most of the chlorine compounds like glucose and fructose, these are the two units, they came out and there was no hydrolysis of any chlorine in the body, so it's a very safe compound. I was asked to work, the work on this particular trianhydro sucrose. What Lemieux has reported in 1959, that if you put 3 tosyl groups at primary positions of sucrose and treat with sodium ethoxide, you get 3, 6, 1 prime 2 and 3 prime 6 prime trianhydrous sucrose, and there is a formation of anhydro across the ring. But later on, Riaz Khan and others, they did the same reaction. They always got 3, 6, 1 prime 4 prime and 3 prime 6 prime anhydro. Nobody ever get this anhydride across the two rings. So I was asked to whether I can be able to make what Limu has reported, by different route. Some senior guys, I asked them, he says, if you are, this project was given to several of the people. If you do it, you are a don. If you are not, then you will be like us. So we did some systematic study here. We made this 3, 6 anhydro and 3 prime 6 prime anhydro and toxin at this position and treated this sodium methoxide with the hope that it can form 1 prime 2 anhydro, but it was never formed. It always gave the 1 prime 4 prime anhydro. Similarly, we have made the 3 prime 6 prime and treated with toxin here with the hope that we get across it. It didn't form, it formed only 1 prime 4 prime anhydro. So all the cases we were getting the anhydro within the ring, not across the ring. So then, we protected these two primary positions and toxin at this position and treated with sodium methoxide. Surprisingly, we got anhydro in which the anhydro ring was across the two sugar agents. So it was very easy at that time because since we got already, so we removed the tritile, put the toxin here and treated with sodium methoxide. So we got what Limu got it, in which there is a 1 prime 2, 3, 6 and 3 prime 6 prime anhydro. So this is the first time we made what Limu has reported at that time. So the conclusion was very simple. We realized that if 3 prime 6 prime anhydro forms faster than 1 prime 2 anhydro ring, because 1 prime is a neopentyl group, it reacts very slow. If once formed this ring, there is a structure of fluoronos folds such a way that the formation of anhydro ring is intact. And therefore, if 1 part anhydro is formed first, then both 3 prime 6 prime anhydro can form concomitantly. So this was a hypothesis we gave. Then we studied its physiochemical properties. What Limu reported was that his melting point is 163, 164, optical rotation is 117, whereas this one has melting point of 194, 196, optical rotation 195, and ours was the melting point was 188, 190, and optical rotation 54. So this is, our one was formed unambiguously, so it didn't match with this. So we don't know what Limu got, but what Limu got we don't know. But what we have done is that we have proved what Limu was supposed to be his anhydro. So that's what I did in London University King's College. So then I went to York in Toronto, Canada for doing my postdoctoral research. And here my association with the project, was such that, I learned a lot how to make the oligosaccharide, how to couple the two sugar units, and what are the different kind of protecting groups, what kind of different coupling reagents which you can get alpha or beta glycosides. So that's what I studied here. The project was such that there was an understanding that N-glycans, nowadays they call, but it was glycoproteins, is formed, by the addition of various sugars on the growing protein unit. The protein is being generated, and different sugar which are present in different species gets attached to the protein, and this is the way the glycoprotein is formed. However, Leloir from Argentina in 1972, he got a Nobel Prize for this work. Paper completely changed the understanding of glycan biosynthesis. What Leloir has said, that the carbohydrate part is not directly attached to the protein, it is formed separately for all the glycoproteins you can think of. And that particular glycoprotein, he called it as a G oligosaccharide. So this oligosaccharide is formed on a dolicol pyrophosphate, which is a lipid, you can see here, by addition of glucanac, mannose, and then at the end there are 3 glucose units, which is alpha 1,3 and alpha 1,2. Once this particular thing is formed, then it gives a signal that your oligosaccharide is ready to attach to the protein. And this is the way the glycoprotein is formed with the basic structure of this oligosaccharide. So this oligosaccharide is now getting attached to the either arginine or serine or threonine of the growing protein unit, and you can see that there are N-acetyl glucosamine, mannose, and glucose. So the revised pathway is like this. Synthesis of dolicol lipid-linked oligosaccharide is the first form in all the glycoproteins biosynthesis first. N-block the transfer of oligosaccharide to the protein, and then the processing of oligosaccharide happens in different species based on the enzymes which are present or the sugars that are present in the molecule. So what happens, this dolicol pyrophosphate, it reacts with UDP glucanac here, and again one, and this is the way the G oligosaccharide is formed after the 3 sugar units or 3 glucose units are attached to the molecule. And I just mentioned like this. Later what happens, once it goes into the system, there are 2 glycosidase enzymes. One is glycosidase glycosidase 1, which cleaves this 1,3, and there is a glycosidase 2 which cleaves this, both of them, and then you get this nascent one. And this is the one which undergoes various degradation of the carbohydrate based on the enzyme, and there was a lot of addition of carbohydrate which is present in different species. To my knowledge, most interesting is that there is not a single glycoprotein in the world where the glucose is present in this structural framework. There is no glucose at all. There is always other molecules like glucanac, galactanac, mannose, arabinose, and all this, but there is no glucose. So the Penmore Corporation, in Canada, they were interested in knowing, what is the role of these 3 glucose units, which are present in the glycoprotein or on the G oligosaccharide. And these are the various glycoproteins present in various species like yeast, insect, non-human mammals, plant, and human. But you see, there is no glucose in it at all. So we were asked to make the trisaccharide. This is the 1,3,1,2, and we put it on a bovine serum albumin, which is a protein, by using a linker, and that's the way wanted to study. The study says that once these 3 glucose units are attached to the G oligosaccharide, it gets a signal that your G oligosaccharide is ready, then end block it attached to the protein, and once that is done, the role of the glucose units are over, and then there are enzymes are there, it glues and forms that nascent, whatever the parent glycoprotein. So this is the work I did. It was very important because I learned how to do the coupling reaction, how to do the temporary rotating group, the permanent rotating groups, and those kind of things, which was very useful when I came to NCL and did a lot of work on oligosaccharide synthesis based on the fundamentals I learned from this work. Research performed at CSIR and NCL. When I joined NCL, Dr. Ramarao's group, he was making adramycin, which is a very potent anti-cancer compound. And in adramycin, at the green tea, there is a glycone, and on that glycone was present a sugar. They call it as donosamine. So since I was a sugar man, so he asked me whether I can make donosamine so that we can hook it to the glycone part, the tetracycline part, and make the complete synthesis of adramycin. So I made it, and this was very useful in completing the total synthesis of adramycin. I don't want to stop because this is what was done a long time ago. And then what happened, I went to IICT along with him, and there we started working on mycobacterium species. The Mycobacterium tuberculosis, Mycobacterium leprae, and Mycobacterium other AVM and all those things are the causative agents for different kind of diseases. And the idea is like this: if you look at the cell wall surface of suppose tuberculosis, for instance, then there are a lot of sugars which are attached on the surface, and they are the ones which are looked by the antibodies. These are the ones which have been recognized by the antibodies. So they have a lot of importance if you want to generate a vaccine, because that's the way. If you can create those kind of sugars, put it on some protein and give, there is a possibility that it can be seen by the antibodies. You can generate antibodies and you can make a vaccine. So work was done by Professor Buchanan from Colorado. He took the trisaccharide unit of Mycobacterium tuberculosis as well as Mycobacterium leprae and put it on this one. Unfortunately, we didn't get the kind of immunity we want when you generate this kind of vaccines. The immunity was from 0 to 60%, not much. Even the BCG injection normally we give at the very young age, also the protection is only from 0 to 80%. So the work on Mycobacterium tuberculosis and leprosy was based on the sugars they isolated from the phenolic glycolipid was not good, So we also make some of the things about half a dozen molecules, but when we realize that there is not much of fun doing it, so we land up only making carbohydrates only. The same thing like that I mentioned here is a major killer. Tuberculosis, mycotuberculosis is the causative agent for tuberculosis. The cell wall surface of this one contains different oligosaccharides. Since the oligosaccharides are outside the cell, they are recognized first and therefore useful for biosynthetic pathway studies as well as for anti-drug TB development. Now, what happens? The phenolic Mycobacterium tuberculosis is not very good. So there were 2 other major oligosaccharides. I want to go back. I'm going up. Yeah. This is a cell wall of Mycobacterium tuberculosis, and the two important one which have now been recognized very lately is lipoarabinomannan, which is here, and arabinogalactan, which is here. So these are the 2 things. They are involved, or they are necessary for the biosynthesis of tuberculosis, or survival of tuberculosis, and by interfering the biosynthesis of AM and AG, is the idealistic approach to discover the anti-TB drugs. So for example, ethambutol, which was discovered 35 years ago, interferes with the biosynthesis of arabinose of lamb and AG, and this is the way it kills tuberculosis bacteria. So these are the different motifs of arabinose. E, A, C, D, and so this is C is a carbon, instead of oxygen we put carbon, and then this is D. This is the first one we synthesized in the first time in the world, and this was done by Hota Srinivas. And I just want to tell you that making sugar units from the pyranose ring is very the pyranose ring is very simple because it has got rich structure. In case of furanose, like we are talking here, this arabinose units, it's very difficult because there is no standard method of making alpha or beta glycoside. You have to use different methods like Inimuse method or Fraser-Leeds method or Schmidt's method and see whether you get good selectivity as far as the glycosylation is concerned. And these are the ones which I've mentioned, these are the one phenolic and didn't show much of antigenic activity, so we landed doing this. When I was there, I approached some people that whether we have got sugar signal, whether they can study is vaccination or as antigenic determinants, whether we can do it, but not much interest was shown. So we landed up making only these molecules. In ICT, we made several of these natural products for total synthesis. I don't want to go into details because these are all published work. But if you look at it, they are very complex structures, and there are a lot of asymmetric centers present here. Some of them lost about 20-25 steps. And this is the way we made several compounds in NCL also. If you look at it, some of these molecules, whose total synthesis was done in NCL. There are also a lot of asymmetric centers are there. These are some more molecules. I think this is the one which is interesting. It's called susticane. We made this molecule because there is a professor in Italy and in UK. They were working on the physiochemical properties of this molecule. So we gave them, and they studied the molecule at various temperatures below 0, minus 273 and all those things. So they were able to collect the data. I don't want to go into details of it because it's a very physical paper, so I don't understand much of it. So this is the work I did it for basic sciences, and there was a story I think I must have told you why I really got involved with the pharmaceutical industry. I think for the young people, I think to repeat it will be beneficial. I was sitting in IICT in my room. There was a call from the security officer that there was an old man, a lady, and a small child wanted to see me. I said, send them. They said, no, they want that I should meet them in the office. So I went there, and there was this old man of about 60s, and there is his daughter maybe in 30s, and there's a small child, infant, maybe about a year old. What they said that, this, my daughter married somebody, and he went to Nigeria to do the work, job work. And he was not keeping well, so they deported him back to India. So he came to India and was still not well, so they took him to All India Institute of Medical Sciences, tested his blood, and he was suffering from AIDS. And he died few months later. But the doctor, told from AIMS told that his daughter and his grandchildren also should undergo the testing. So they did it, and both of them were HIV positive. This is one part of the story. The second part of the story was like this, that in 1987, I was invited by Doordarshan in their Good Morning show. It used to be there early in the morning in 87, something like that. And there you have got lot of things going on there, and they ask you question as a chief guest or something like that, and asked me at the end, what is the new thing which is going on? Then I told him that AIDS is going to be a problem for India, although people say that in India we won't get AIDS because this is only confined to homosexual, and but it's not true. So since this Boros welcome AZT was approved, so since it is a nucleoside and I'm a carbohydrate, so I made some few grams of this material in case, suppose it breaks, HIV breaks, then you should have a technology to make the AZT for people. This is one thing. This particular doctor probably read or was witnessed my interview, and he told this gentleman, this old man, that there's only one person in India, and he's a IIT Hyderabad, who can give you this AZT. And therefore they came to see me. I told them, that there's no way I can give. We have not done any toxicity, we have not done any formulation, we have not done the bioequivalence studies, not done the clinical trials, and not gone to regulatory affairs and all this. So there is no way I can give. Of course, they left. Once they left, then I realized, that what we were doing at that time was only to do the science, publications, and that's it. That's the way, my work was going on. But that day onwards, I thought that I must do something for pharma industry because we have knowledge and we can do something better for the industry. So we were very lucky that so many companies at that time sponsored projects to us, particularly Sipla. They gave us various projects, and based on that, they made me also the board member of the as a board member of their company. So, application science for pharmaceutical industry. That's now going to be the last chapter of my talk. We realized, because I'm part of both academia as well as industry, for many years in the academic and pharma industry have had a symbiotic coexistence. They were there in the system. Academic mission was that you do the advancement of frontier science, disseminate the knowledge through publication and scientific tools to undertake challenges, but they oppose to clinical application. Whereas the pharma industry developed new medicine, protect IP, intellectual property, maintain profit margin for the shareholders, and immediate impact on the patient lives. So that's their portfolio. So industry invites academics as guest workers, speakers, or consultants in different capacities. But we were at advantage in industry because we had the best of both the worlds, access to the academic research, which is publication, as well as pursuing our objective of drug discovery and development. So this is actually the working in science for academic versus industry. So we started getting a lot of projects, and this is the work I did it in NCL, that's what I wanted to tell you. This is the molecule, developed by Apix Pharmaceuticals, and it is a contrast agent for MRI detection. If you look at it, it has so many peptides, amino acid as a peptide. And since we didn't have the solid phase synthesis facility in NCL at that time, so this was done by solution chemistry. So we were making one and adding one like that. And then later on, we also did this. This is kind of a profiling kind of thing. And there is a gadolinium, in the center of it. And then we hooked it through the next slide. Through this pentafluoro phenoxy derivative or phenol derivative, and this is the way we made the entire contrast agent. And this is now in market actually. We developed the technology and gave it to them, and they were making this product. This product is there in the market. This is the pig model of thrombus. You can't detect anything here. Once the contrast region is given, we were able to see that there is a clot here. And quite important, because I don't have the figure, this particular thing doesn't give only the two-dimensional structure of the plot, it gives a three-dimensional structure. So you know what is going to be is width rather than a length and breadth. Is the depth, So this is the way. There are so many of examples they have shown us that the molecule is working so well. So this is the work we did it in NCL. Additional work which we did in NCL was based on the chirality. The chiral information started by thalidomide tragedy. In 1957, the German company, introduced thalidomide for morning sickness to the pregnant woman. 10,000 infants were affected because this was given as a recipe mixture of S and R. And S is the one which is teratogenic, gives the children born without limbs. So it affected a lot of people. I suppose about 10,000 now. At that time, it was realized that the chirality has got importance from the biological point of view. So I was in Sepracor, at that time, my friend Chris and I invited me to do a presentation, and I realized that Sepracor is doing the same thing. There are about 1,700 at that time racemic compounds which are introduced into the market. So he said that their company is going to do the separation of both the isomers or whatever the inosomers and see which one is active and whether we can have the same activity with half the dosage level, because unwanted we are going to remove it. So that idea was in my mind, and in 2000, Mr. Satish Mehta and Mr. Khanna from MQR, where I'm working today, they came to us and said that we wanted to start something new in API area. Because that company was already doing formulation, they were buying the API or the bulk drug from outside, they were doing the formulation and selling it. So they were always never used to be number one because they had to buy API from somebody else. So they wanted to create the facility to make the bulk drug and then use it for formulation. So I propose, that this is the area they should do because nobody has done it so far, where you can separate the chiral racemic compound and get a chirality products, as single isomers and see whether they can act out there. They like this particular idea, and the molecule which we identified at the time was amlodipine. This was done in NCL and combined. So amlodipine has got a carbon center. We separated by using the resolution process, R and S, and we found out that the activities there always in S, R has got no activity whatsoever. So this is for calcium channel blocker, half the racemic dose, less metabolic load, consistent pharmacokinetics, prevent the accumulation of R-amlodipine. And R-amlodipine we know that causes pedal edema, that is swelling of your legs. So once you remove that, if you take only S-amlodipine, then this issue of swelling of your legs was not there. And people, girls who use skirts, for them it was very difficult. So now once you have got this S1, they don't have to really worry about that. So, also we did in NCL metoprolol. This was also introduced as a racemate. We made S metoprolol by using the parasynthetase epichlorohydrin, and again, this provides a beta blocker component only. The half the doses, avoiding beta blocker blocking component, and there are so many other advantages with chiral metoprolol rather than the racemate one. We also did acetanolol, which is also a beta blocker. Same advantages, half the dose, and it has got almost the same activity. And I want to tell you that amlodipine today is about 100 crore product for the company, and metoprolol is selling around 50 crores, and atenolol is not introduced in India, but we are introduced in Korea. So we do a lot of business with Adenol in this. So I just want to thank Dr. Joshi because all the work it has been done under his supervision, Dr. Ramesh Joshi. Yeah, these are the work which we continue doing it in MQL. Like rabiprazole is a proton pumping agent. We see that R is the active one, with the X is the active one, and then we remove the less one, inactive. And again, dose level is less. S-pantoprazole, again there's a target center. Just like S-omeprazole, we went S-pantoprazole, and same activity is half the dose and so on. Dextromethorphan, this is a very powerful anti-inflammatory drug, again, half the dose in pain. I take Infen when there is a real pain, it goes like that. Then dextromethorphan, which is very important because you remember that when you take the racemic ibuprofen, R and S, the S is the one which has the activity, R doesn't metabolize at all. So what R has to do in the body is that convert into S and then it metabolizes. So R remains in your body for some time. It's because it becomes a metabolic load for you. So if you remove the R, then the S reacts very fast, and it comes out very fast also. Etodolac, this is also an anti-inflammatory drug, doing quite well. Sibrutamine, this is for anti-obesity. We converted into, we separated both R and S and introduced R. Then R ondansetron for nausea and vomiting. Again, converted the racemic into a chiral bond. Zopiclone, of course, this was known. This was developed by Cepracor, so we also did it. So these are some drugs, which we did it, and developed this technology. And today, I am very happy to say that we have about 110 drug master files which we have filed in US FDA. That means that we have got 110 technologies which are present with us. Drug master file is that you make a file with the API with all the stability data, then your bioequivalent studies, and so many impurity levels low and so many things, and then you file this particular dossier and then you get FDA approval. Then you can sell it anywhere in USA and other countries. So this is the way we do a lot of work, and these are some of the examples. See, there are drugs which are inspired by natural products. So different sources of natural products use different kind of very exotic structures of natural products, which we cannot think of. Even if you sit down, you will not be able to see that this is the molecule one can make. For example, from plant, you get morphine for use for pain. From marine, you get a product like eribulin, anti-cancer. From frog, you get acetonide, anti-diabetic. Is a very powerful compound, acetonide. It has got 32 amino acid residues, but very powerful compound. Then from microorganism is vancomycin, is also antibacterial. See, these are some of the advantages and disadvantages. I don't want to go into detail, but let's go further. This is interesting. Progesterone is a natural product, and this is dihydrogesterone. This is given for threatened and recurrent miscarriages during pregnancy. It can stop it. But nowadays what is happening is that the young pregnant women, they are very worried about miscarriage. They start taking dihydrogesterone from the day one. Actually, it's really not required. It has got some other properties as I mentioned here. Compared to progesterone, this is 20 times more potent, this molecule, and 5.6 times higher bioavailability compared to progesterone. You look at the structure of progesterone and dydrosterone, only the centers 10 and 9 are reversed. And also there is an addition of a double bond here. To make it by conventional method, this particular molecule is a Herculean task. You will not be able to develop any technology out of it. So how do you get this kind of things? We have to use the photochemical reactions on a large scale, and I will tell you what scale we do it. This is our brand on dydrosterone. It's called Dydrofam. So the properties are like the spatial orientation, which hydrogen atom present at 9 I mentioned to you to be beta, and the methyl group should be alpha. This is the requirement. These juxtaposition of hydrogen and methyl are opposite to those of progesterone, and therefore they bend in a spatial geometry, and the angle between the AB and CD ring, this is AB and CD ring, should be 60 degrees. That's very critical. Then only you get the activity. So these structural features of dydrosterone compared to progesterone make it orally more active and metabolically more stable. So these are the requirements. So what we do is that we make a diene. I mean, these are not very difficult science. You make the diene here, and we do the photon bombardment at a lower wavelength. So there is a retro Diels order, and you get a triene here. And then you bombard it again with a very high wavelength. Then again the Diels order takes place, and you get the concomitant epimerization at 10 and 9. So that's what we wanted. But the disadvantage of this reaction is that 50% it goes back to the starting material. But that also can form. So we separate it out by prepared to chromatography, or sometimes now we do the crystallization, and the deprotection you get is. The Abbott people started saying that our product may not be same as their product because this angle is very important. So I rushed to Rajesh Gonare and I told him that this is the innovator sample and this is our sample. Why don't you do the single electric crystal and show that both of them are same? And he did the outstanding work. If you see it, the yellow one, the green one is ours, and the white one is the innovator sample. So, thanks, Gonare. You paid for it. This is another molecule which are based on natural product. This is eribulin. Eribulin is for metastatic cancer. This is the best one, developed by Kishi at CSIR company. And he needs about 50 to 70 steps to complete the synthesis. Now we can do it in less than 40 steps. One of the most expensive drug, and the dosage is 1 milligram per 2 ml. While playing tennis, I hurt myself. I'm 73 years old.
I can sit down and
Sorry about that. So, I'm very happy to say we are the first generic company in the world to introduce eribulin into the market. Eribulin actually is a part of a marine natural product called helichondrine. And Kishi reported that this part is enough for its activity. You don't need to have the complete molecule. So this is our brand. There are 18 target centers, 5 million different isomers possible. We have to make only one. Installation target center by osmotic synthesis is difficult. So most of the chiral centers are derived from carbohydrates. So we made some of these intermediate, but now they are available in India as well as in China. These 3 units are there, and these 3 units are responsible for the making of ergotamine mesylate. So I don't want to go into details about it, but just to show what kind of new science which we have to do it. We never heard of it. This kind of science is possible in the industrial scale. At this time. It goes down, but not here. Anyway, I'll finish talking quickly. So you do the NHK reaction to do the coupling of this vinyl iodide with this aldehyde, and this is done in the presence of this chiral ligand. So you get the rights to the chemistry here. You mesylate it, and then you of course form the ring here. This is the way, and with diabol you remove the fluoride. So. And then there is a second, third intermediate here. You do the Julia coupling reaction here to this one, and again you DMP oxidize into aldehyde, and again do the NHK reaction where you get if you don't use any chiral ligand, you get a mixture of here. That's not important because we are going to oxidize the center anyway. So this was then treated with samarium iodide to remove this phenyl sulfonate. So you get this intermediate here. So you do the oxidation, and unfortunately the structure has not come at the end, So, but I'll try to explain it. If you treat with tetraglutamine fluoride, all the TBS groups are gone, and it's basic enough. The hydroxy group here then undergoes 1,4 addition on this one. So you get here. This is the one. I don't think I'll be able to write. No, it's just this last 50. I will do this. And then what happens? These 2 hydroxyl groups and this ketone, they form a ketal in the presence of PPTS. So you get this particular ring structure. Then you, of course you have to make only the amine here. So this is a triplet, and then you get a 0. And the last structure which is not there is just a amine. So this is the way we make ergotamine. The batch size is about 25 grams. The cost of this one, if you do under the GMP, is about $50 per milligram. If you do non-GMP, it's $30 a milligram. So it's a very expensive, expensive API. This is testosterone, which is used for pulmonary arterial hypertension. This is synthetic analog of prostacyclin, and its formulation is very unique. You have to do the inhalation. Synthetic challenges 5 centers, and you are using the asymmetric Paulson-Kahn reaction for the critical steps. This is we do this not in India but in UK, because we have a company in UK called Telomeres, so they are the one who introduced this one in UK. If you want to do Paulson-Kahn reaction, this is the intermediates. This is suppose starting material. Cobalt carbonyl, you can get this tricyclic ring, which you will want it. And this is the racemic one. So if you want to introduce a chiral center here, what we did was that we introduced some chirality here and did the Paulson-Kahn reaction, for example, here. Then, when we did it, we got the chiral center fixed here, and which was very critical. So what we did was that we prepared this intermediate, in which there is a chiral center here. We did the Paulson-Kahn reaction with cobalt octacarbonyl. We got this particular compound in which we were able to fix this chiral center because of the presence of this. And now if you look at the molecule, we don't need it, so we did the hydrogenation to remove this benzenic hydroxyl. At the same time, we also reduce this one. In this case, when you reduce it, you get cis here, but there was a problem with this particular center, which was in mixture. Very interesting things happen. You have to now reduce this carbonyl here. And this is a mixture of R and S, beta and alpha. What was realized that if the hydrogen is alpha, the reduction of sodium borohydride takes place so fast, whereas the one, if it is beta, then it doesn't take place at all, very slow. So how do we do it? We added sodium hydroxide in sodium borohydride. The one which is alpha went, and the one which is beta isomerized to alpha because there is a sodium hydroxide, and also it went to give the right sodium state. So this is the way we were able to complete the synthesis of this molecule. There are a lot of good things we can learn from here. This is the last one. Trabectedine. Trabectedine is also a very potent marine natural product. It's been approved by FDA. Its natural availability is about 1 gram for 1 ton of tunicate. This was actually synthesized by Corey, and Corey's intermediate was available from China. So we little bit modified Corey's synthesis because it should suit our process, yields and quality and those kind of things. Because he made it only the last reaction, 3 reactions he did on 1 milligram compound. It just, I don't know. So this is the synthesis. We first make the ester, and then with palladium we remove the allyl. It forms immediately the ketone. And with selenium, benzoselenium anhydride, we introduce the OH group. And when you treat with triptylic anhydride and a little bit of base, it forms a double bond here, right? I'm not here on the structure. It forms a double bond here. And then we treat with another base. This protecting groups, and then goes, it gives a nascent sulfur, that undergoes 1,4 addition here, and you get this bicyclic compound. And then of course with tributylene hydride, we remove the allyloxy, use this method of pyridine benzaldehyde, 4th position, pyridine aldehyde, and this phenyl sulfonate to get the ketone, and then you do the Stecker reaction to complete the total synthesis of tributylene. So this one we made up a few grams, and work is going on. We have to do some bio equivalent studies as well as CT. Once that is done, we will be able to introduce this product in the market. See, and this is 50 years of work, and this is only part of what I've done. There are a lot of people, who are involved in framing me what I am today. First, my teachers, Professor Bhagwan, Professor Leslie Hawk, Professor Richardson, and Aspiron, where I did my PhD. Then mentors from academia like Professor Rama Rao, M. Sharma, Mashilka, Paul Swami, Dr. Shiva, K. Ganesh. They were also responsible for my progress. Then my mentor from industrial like Mr. Satish Mehta, Dr. Hamid, Prashant Tiwari, Rama Mukherjee, Anand Burman, M. K. Chandavarkar, Glenn Saldana, Sudarshan Arora, and Abdul Khanna. They were all, gave a lot of funding to us when I was in NCL. And my colleagues from NCL, Dr. Joshi, M. Deshmukh, Dr. Ramana, Mahatma Kalpote, Jawan, Bhanu, and Dr. B. H. Deshpande. Last but not the least, the outstanding research scholars I have. I just want to tell, there are a lot of stories, when you have so many, hundreds of PhD students, then a lot of things happen. So I'll just tell one small story and end it. There was one student who was facing his viva for his PhD, and he did some work on swan oxidation. So the examiner asked that you have done swan oxidation. Is there any other method you can do the oxidation? He said, of course you can do it. He said, tell me. I don't know. So
बहुत बहुत धन्यवाद। और इसी बीच हमारे निदेशक महोदय आदरणीय डॉक्टर लेले सर यहाँ उपस्थित हुए हैं। आपकी गरिमामय उपस्थिति को प्रणाम करती हूँ और सादर आपको मंच पे आमंत्रित करती हूँ। जोरदार तालियों के बीच। और जो आदरणीय सर ने अभी डॉक्टर गुर्जर सर ने अपनी विज्ञान यात्रा के बारे में जो रोचक जानकारी दी, उसे कहने के लिए मेरे पास शब्द नहीं बचे, निशब्द हूँ मैं। और मतलब वो बताता है ये इनका जो इतना 50 वर्षों का महत्वपूर्ण जो अनुसंधान है, ये पता चलता है कि सर के पास देने के लिए बहुत कुछ है, लेने वालों को ही अपनी तैयारी रखनी है कि आप कितना ले सकते हैं। तो आपकी प्रेरणा, आपकी पॉजिटिव पर्सनैलिटी, आपका पॉजिटिव एरा, आपका जो पॉजिटिव ओरा है वो हम सबको हमेशा प्रेरित करता रहेगा। मैं पूरे एनसीएल परिवार की ओर से शुभकामना देती हूँ। आपका स्वास्थ्य हमेशा अच्छा रहे और आप इसी तरह से काम में अनवरत लगे रहें, लोगों को प्रेरणा देते रहें, मार्गदर्शन देते रहें। और आपने संपूर्ण मानवता के लिए, मैनकाइंड के लिए जो भी आपने रिसर्च वर्क किया है, दवाइयों के क्षेत्र में महत्वपूर्ण अनुसंधान दिया है, वो निरंतर जारी रहे और हम सब उसके लिए हमेशा गौरवान्वित रहेंगे आप पर। आपके द्वारा ऐसी कई 100 और महत्वपूर्ण खोजे हुए यह शुभकामना, और आपके जैसे कम से कम 100-200 साइंटिस्ट और ज़्यादा आपसे प्रेरित होकर आपके जैसे उपलब्धियां अर्जित करें, यह कामना करती हूँ। सर बहुत बहुत धन्यवाद, मेरे पास शब्द नहीं है, तो बहुत बहुत धन्यवाद। और इसी बीच मैं लेले सर को आमंत्रित करना चाहूँगी कि इस अवसर पर 2 शब्द कह के हमें अनुग्रहित करें।
First of all, Dr. Gurjas, what can I say? So many thanks to you for accepting our invitation and coming here and delivering this very important talk that we conduct at a regular frequency. First of all, I hope you're okay.
Yeah, I'm okay.
The passion with which you talk, despite the pain, you still wanted to continue and finish the talk. I was sitting there and getting very worried, but you wanted to still continue.
I think you have to blame, Mahesh Kulkarni, because he's the one who really hit me.
So Mahesh, there is a penalty, and he will pronounce what the penalty is. So thank you so much, Dr. Gurjar. Your association with NCL has always been extremely valuable. I just hope that the royalty that we get by your doing such phenomenal work and transferring technologies to companies like Encure, I wish there was an additional royalty component of moving you to Encure. And that would be so invaluable for us and absolutely perpetual. We are very grateful for you, not just for the work that you have done at NCL and shown us the way, but also the fact that the ethos of NCL, of not just doing phenomenal scientific research work, but always having a goal of what that research is meant for, and how that research translates into value, not just monetary value, but value that saving lives is a value that is invaluable. So having that very sharp focus in your research always is a great motivation for us. It is what NCL is all about, and I hope that this tradition continues at NCL and we have many more Dr. Gurjas coming out of this very wonderful laboratory. I honestly really don't have much to say. Dr. Swati had already said some excellent words, and I hope that you get out of this pain very quickly. We see you more often on the tennis courts, and we also see you more often in NCL. We would like to have your guidance, sir, for where organic chemistry will be headed in the future, because I think a lot of things have changed. And like the way you shaped the research of companies like Encure by bringing in a new dimension, which at that time was chiral chemistry, we are looking for that new dimension that NCL will add and steer the way of the pharmaceutical companies in India. We are aware of the many directions, the new directions that are emerging out, but your guidance to us would be very, very invaluable. So we look forward to receiving that from you. Thank you so much.
बहुत बहुत धन्यवाद डॉक्टर लीले सर, आपकी प्रेरणादायी और सरल सुंदर शब्दों के लिए, प्रेरणा देने के लिए बहुत बहुत धन्यवाद। और कार्यक्रम को आगे बढ़ाते हुए मैं माननीय लीले सर से निवेदन करना चाहूँगी कि कृपया इस अवसर पर हमारे मुख्य अतिथि जी को मेमेंटो देकर सम्मानित करें। और क्वेश्चन आंसर सेशन हम लोग स्किप करना चाहेंगे। पूरे एनसीएल परिवार की ओर से सत्कार और शुभकामनाएं स्वीकार करिए सर, और आपका आशीर्वाद सदा बना रहे। और इसी के साथ हम बढ़ते हैं कार्यक्रम के अंतिम चरण में। मैं डॉक्टर मोनिशा को सादर आमंत्रित करना चाहूँगी वोट ऑफ थैंक्स के लिए।
On behalf of the organizing team, it's my pleasant privilege to propose this vote of thanks. First and foremost, I would most sincerely like to thank our speaker for today, Dr. M.K. Gurjar, for taking us through this phenomenal journey of his 50 years, delivering such an inspiring talk in spite of severe personal discomfort towards the end. But yeah, so thank you very much. Also our director, Dr. Ashish Lele, for his continued support, and also the NCL Research Foundation. Thanks to our Organic Chemistry Division Chairperson, Dr. Ramana, the OCD office staff, our Director's office staff, and also we would like to thank Dr. Swati ji for anchoring today's event and making it special. We especially thank the events management team, the entire team. There are so many people who are involved in this and contributing towards organizing this